So, um, my project is predicting mature microRNAs from pre-microRNAs using machine learning, and my name is Pavan Angara, so let's get into it. So, microRNA is a tiny little piece of RNA that can be used for gene regulation in the body. So it figures out what kind of proteins we're showing, what kind of proteins we're not, and a lot of other things. And it's important. MicroRNAs are important because they can lead to major breakthroughs in agriculture and medicine. They can help prevent diseases in plants and animals and humans. Um, and at Cornell, we're particularly interested in plant microRNAs, so I was inspired by the Perry Lab of bioinformatics, which I work at, to pursue this project. On the left, we see a diagram of just the microRNA life cycle. So we start with a pri-microRNA, that gets cleaved to become a pre-microRNA, and then that then gets cleaved again to become a microRNA. We're particularly interested in this part here, which is going from a pre-microRNA to a microRNA. Um, so there are a lot of machine learning algorithms that exist to try to take a pre-microRNA sequence, genetic sequence, and from that predict what microRNAs might come out of that pre-microRNA sequence. So a few of the ones that I looked at for this project are mature PRED, which uses support vector machine and RNA structural analysis. I also looked at mature Bayes, that uses a naive Bayes classifier to try to figure out microRNAs from pre-microRNAs. And then there's also Mir locator, which is no longer available, which uses decision trees. There's a lot of options, but most of them lack customizability. So in other words, you can't, they've already been trained, you can't figure out, or you can't input new training data sets for most of them. So that's a major setback for a lot of biologists. So we need a prediction program that can, one, work well with smaller sample sizes. Why? Because in the lab, they often get just very few examples of microRNAs. They may be working with rare species of plants and animals, and there's just not that many classified examples that exist in the wild for them to look at. So we need an algorithm that can work with smaller sample sizes. We also need something that can quickly train on new data sets. In the lab, they're constantly comparing and trying to better classify using combinations of data sets. So we need something that doesn't take a lot of time to train on each one. We also need something that can be easily understood by non-computer science researchers and academics so that they can go into the code and change it themselves. I ended up choosing k-nearest neighbors. The basic outline for k-nearest neighbors in this case is that we start with a pre-microRNA, which is about 100 to 160 nucleotides. We then have a function which finds all 20, 21, and 22 nucleotide continuous sequences in there. So pretty much it just looks at every 20 nucleotide continuous sequence in there that could possibly happen. It lists them all, and it does that for 21 and 22 as well. Then it takes the list of all possible microRNAs that could be cleaved from the pre-microRNA, and it does a distance calculation on them from the training set. So it takes the first possible microRNA that we found in the first step, and it compares it to everything in the training set using a distance formula. And then it does that for everything in the training set. So it takes this possible microRNA, compares it to everything in the training set, and then it takes the k closest microRNAs from the training set, and if the majority of those are positive, then we predict that this possible microRNA is positive. We say, yes, it's going to be probably in a microRNA. If the majority are negative, we say the opposite. We say, no, it's not going to be a microRNA. And so at the end, we end up with a list of classified microRNAs for the initial pre-microRNA. The most important part is the distance equation. I chose to use Levenshtein distance. So essentially, Levenshtein distance just calculates the number of changes that need to be made to transform string 1 into string 2. In this case, sequence 1 into sequence 2. And that was used to calculate the distance between two sequences. We gathered data from something called MERBase that has all valid microRNA samples, and negative samples had to be generated because there are very, very few negative examples in the wild. So we generated them by taking random, invalid 21 nucleotide sequence from a pre-microRNA. So we just took a 21 nucleotide sequence from a valid pre-microRNA, and we said, is this valid or not, this pre-microRNA? And if it wasn't, we said, okay, that's a good negative example. We compared our algorithm to existing programs, and not surprisingly, it did not do as well in some respects. So its recall was very high, but its precision was very low. What this means for k-nearest neighbors that we implemented is that it pretty much looks at a massive amount of possible microRNAs. Like I said, all 20 nucleotides, all 21, all 22. And it does the mathematics on those, the distance calculation, but it ends up choosing a lot of false positives. That being said, it has a very high recall because it chooses the right. It almost always will choose the correct valid microRNA, but it also chooses a lot of microRNAs that aren't really microRNAs. So then we looked at the effect of varying the training data set on precision and recall. So we were kind of surprised to find out that it's not necessarily, if you're looking at Arabidopsis thaliana, which is a plant, for example, if you're trying to classify those pre-microRNAs, if you're trying to classify those microRNAs, then you're not necessarily going to just want to train with Arabidopsis thaliana samples. Instead, we found that a mixture of different species worked best. So in this case, we looked at Vitis vinifera, and we found out that a mixture of Arabidopsis thaliana and Vitis vinifera, 50-50 mixture in the training set, actually led to the best results, the highest recall. So the reason for this is most likely that when trained on just Vitis vinifera samples, there was too much there was too much overfitting. So it couldn't generalize to new microRNAs it had never seen before. We also looked at the effect of increasing neighbors on precision and recall. So you can see here that over time, as the number of neighbors increases, it gets to a point where there's a peak, and then after a while it starts to decrease. This makes sense because if you're starting out at the very left side and you have very few neighbors, you're really only checking with one or two other examples, and that's not really a good way to classify anything because you don't have enough information about the situation to classify. Then on the other end of the spectrum, if you're looking at almost all of the samples that we used, you're pretty much just taking a majority vote of the samples, which again isn't really specific to the microRNA that you're looking at, so that's a problem. And then real fast, I'm just going to show you a little bit of the code. So this is pretty much a very long script with a lot of different functions in it. And I want to show real quick just the negative, if I can find it. Maybe I can't find it. Regardless, another thing to show real quick is just the bash output. So here we see a huge ugly thing, but this shows the positive examples that it found and the negative examples that it found. And in this case, it returned this precision and recall, and we do that over a bunch of samples, and then we take the average to figure out the average precision and recall. But yep, that's pretty much the program.